Welcome to CUSABIO. Today, we will introduce you to the Plasmid DNA Purification Maxiprep kit. This kit provides a quick and simple method for isolating high-quality plasmid DNA from bacterial cells. Let's get started. The CUSABIO Plasmid DNA Purification Maxiprep kit is based on a novel ion exchange column purification technology to extract transfection-grade plasmid DNA. This kit not only works quickly and efficiently but also includes a unique endotoxin removal reagent, making it suitable for various downstream applications such as transfection, transformation, lentivirus packaging, CRISPR genome editing, sequencing, restriction endonuclease digestion, PCR, in vitro transcription translation, and more. Using a phenol chloroform-free method and just a 3-step flow-through column process, you can purify up to 1.5 milligrams of plasmid DNA from 150 to 300 milliliters of bacterial culture. This experiment uses the alkaline lysis method to extract plasmid DNA, which includes 3 basic steps: preparation of a large number of bacterial cells, collection and lysis of the cells, and isolation and purification of plasmid DNA. The kit includes the following components: S1 buffer, S2 buffer, S3 buffer, RNA-seA, ER buffer, 2 bottles of wash buffer, elution buffer, TE buffer, and 5 plasmid purification Maxiprep columns. Now, let's go through the experimental steps one by one. Step 1. Bacterial culture. Begin by inoculating 50 ml of glycerol stock or a single colony into 3 milliliters of LB liquid medium containing the appropriate antibiotics. Incubate overnight at 37 degrees Celsius with shaking at 220 rpm. Next, transfer 200 ml of the overnight culture into 200 milliliters of LB medium with antibiotics and incubate overnight under the same conditions. Step 2. Harvesting bacterial cells. Pour the 200 milliliters culture into a 500 milliliters collection bottle and centrifuge at 8000 rpm for 3 minutes at room temperature. Discard the supernatant, and invert the bottle on lab paper to remove any remaining supernatant. Step 3. Resuspending bacterial pellet. Add 10 milliliters of S1 buffer to the pellet and use a pipette to resuspend the cells completely. Split the resuspended cells into 2 50 milliliter centrifuge tubes. Step 4. Cell lysis. Add 5 milliliters of S2 buffer to each tube. Close the lids and gently invert the tubes 8 to 10 times to fully lyse the cells. After 5 minutes, add 5 milliliters of S3 buffer to each tube. Mix gently until white flocculent precipitates form. Let the mixture stand for 5 minutes, then centrifuge at 4 degrees Celsius, 12,000 rpm for 20 minutes. Step 5. Filtering the supernatant. Filter the supernatant using a 20 milliliter syringe and a 0.45 micrometers filter into a 50 milliliter centrifuge tube. Step 6. Column preparation. Label each column with the project name and secure them sequentially on a rack. Remove the caps and allow the storage solution to drain naturally. Next, fill the columns with pure water to wash the resin thoroughly. Finally, equilibrate the resin by filling the columns with wash buffer. Step 7. Endotoxin removal. After filtering the supernatant, add 3 milliliters of ER buffer to each centrifuge tube. Mix well and incubate at 4 degrees Celsius for 30 minutes to remove endotoxins from the plasmid supernatant. Then, pour the supernatant into the column and allow it to pass through by gravity 3 times. Step 8. Washing and elution. Wash the column twice with wash buffer. Then, add 10 milliliters of elution buffer to the column and collect the eluate in sterile centrifuge tubes. Step 9. Precipitating and washing plasmid DNA. Add 5 milliliters of isopropanol to 10 milliliters of the eluate, mix well, and incubate at -20 degrees Celsius for 30 minutes. Centrifuge at 4 degrees Celsius, 12,000 rpm for 15 minutes, then carefully discard the supernatant. Wash the pellet with 1 milliliter of 70% ethanol and centrifuge at 12,000 rpm for 10 minutes. Discard the supernatant and repeat the wash with another 1 milliliter of 70% ethanol. Centrifuge at 12,000 rpm for 5 minutes. Remove the supernatant with a pipette and air dry the pellet in a 60 degrees Celsius oven for 10 minutes, ensuring no ethanol residue remains. Step 10. Dissolving and storing plasmid DNA. Dissolve the plasmid DNA in 200-500 ml of endotoxin-free water. Store at 4 degrees Celsius for short term, or -20 degrees Celsius for long term. Step 11. Plasmid quality control. Use a nanodrop to measure the OD values of the plasmid, recording concentration, volume, OD260 OD280, and OD260 OD230. Then, assess the purity of the plasmid and check for RNA contamination using agarose gel electrophoresis. Here are some common questions and their answers regarding the experiment. Thank you for watching. We hope this video helps you better understand and use the CUSABIO Plasmid DNA Purification Maxiprep Kit. For more information, visit our website or contact us.